Perfect. And then I will go ahead and introduce you. So, Luis Miguel Oliveira is an expert in the field of Parkinson's disease research and investment with more than 15 years of experience in the neurologic disease space. Currently, he holds the position of Senior Associate Director of Research Programs at the Michael J. Fox Foundation, where he leads various efforts for the therapeutic and biomarker development of priority genetic targets for Parkinson's disease, accounting for over 30 million in executed projects. Additionally, Dr. Oliveira is the founder of the VAT PACE Alliance, an organization aimed at connecting families affected by rare diseases related to VAT PACE with the research and medical communities for further advancing therapeutic development. With strong commitment to advance novel treatments from the bench to bedside, Dr. Oliveira serves as a review editor in Frontiers Neurology, Neuroscience and Psychiatry, and is on an investment advisory panel for Portugal Ventures, where he leverages his expertise to guide investment decisions in promising ventures. Recently, Dr. Oliveira had an integral contribution to a landmark study that validated the first biomarker for Parkinson's disease diagnosis. This significant achievement was reported in Lancet Neurology with broader international media coverage and is changing the Parkinson's research and drug development field by enabling an objective comparison of the disease based on its biological features. Dr. Oliveira's educational background includes a degree in biochemistry from the Faculty of Sciences of Lisbon University, Portugal. He further pursued his academic journey with a PhD in molecular biophysics from the same institution. During his postdoctoral research experience, he conducted his studies at renowned institutions such as the Max Planck Institute for Biophysical Chemistry in Germany and Columbia University in New York. A biological definition and integrated staging system for Parkinson's disease is what Dr. Oliveira is presenting to us today. So, with that, I'll ask you to welcome Dr. Oliveira with me.
Thank you so much for this introduction. Hi, everyone. It's really a pleasure to talk to you today. And I want to thank the organizers for the interest in this topic. I'll be talking about some recent developments that we're working on at the Fox Foundation regarding the Parkinson's disease research field that I believe are going to have and are already having a profound impact on clinical development and novel treatments for this disease. But to be more accurate, in fact, the title of my presentation should refer to synuclein or neuronal synuclein disease. And hopefully throughout my presentation, you'll join me in appreciating this new concept. Quick slide on my disclosures. I really have nothing to declare. And I want to start, by showing sort of the original descriptions of Parkinson's disease, which included these four features of bradykinesia, rigidity, tremor, and abnormal gait and posture. And from the beginning, bradykinesia, the slow and low amplitude of movement, was always the required motor movement. But broadly speaking, these symptoms are what is referred to as Parkinsonism. Now, idiopathic Parkinson's is really just one possibility. And in fact, when clinicians see patients for the first time, they have to figure out what the heck that clinical Parkinsonism means, as it can be any of these disorders that result, especially early in disease manifestation, in a similar clinical presentation. When I think about the milestones in our understanding of Parkinson's disease, starting way back in the 1800s with the assay of the shaking palsy, transitioning to 1912, where we got the first drawings of Lewy bodies within the substantia nigra by Dr. Lewy, followed by, I think, the very important moment where our first medications came into play with carbidopa and levodopa, which actually allowed us to transition this from describable disease to treatable disease. In 1998, when Lewy bodies were first identified as having alpha-synuclein as their core protein. And now I'm adding 2023 as a sort of the next big biological milestone when we transition our definition of Parkinson's from being defined by bradykinesia plus rigidity and tremor to a more biologically based definition and staging. And hopefully our next big milestone is going to be the time where we can update our 1967 New England Journal of Medicine paper with the first successful disease-modifying therapy for these patients. So how did we do this? Well, we've known for over 20 years now that alpha-synuclein misfolds and accumulates in the brain of individuals with PD, but it is known that these synuclein aggregates travel everywhere in the body through CSF, blood. They are present in many different tissues and even secreted in urine and saliva. But the amount is so small that it was really impossible to detect them in a robust way by any standard techniques. So in 2018, we started working on a new technology stemming from the prions disease field, where we leverage a biophysical property of these amyloid proteins. These proteins are able to seed and template their pathological structure, converting normal monomeric protein into aggregates. And so facilitating this process with many cycles of seeding and fragmentation, we can amplify the levels of aggregates in the sample in order to detect them. Importantly, if the biological sample does not have the presence of pathological synuclein to begin with, which is expected in a healthy control, then this process does not occur and no signal is detected. And what you can see in this graph in the right, is that this test really gives a robust separation of individuals with PD versus controls, and even allows the separation of two different synucleinopathies, Parkinson's disease and multiple system atrophy, due to robust differences in the kinetics of the amplification reaction. And this is, essentially the core of the Lancet paper that was referred in the introduction. After many years of working on optimizing and scaling up this assay, we finally validated this test in a large natural history study sponsored by the Fox Foundation called the PPMI, or the Parkinson's Progression Markers Initiative, with thousands of individuals that who are enrolled in the study, and from who we have deep clinical and biomarker characterization. And that study, was published then this year in the Lancet Neurology. So in that study, we confirmed, well, first and foremost, we confirmed the high diagnostic accuracy for sporadic PD and healthy controls with over 90% specificity and sensitivity. And then we identified two very interesting observations. One, we observed that in LRRK2 PD, there was a high variability in the rates of positive alpha-synuclein SAA results, depending on olfactory performance. We observed that many individuals with LRRK2 PD were actually negative for alpha-synuclein SAA, and this was strongly associated with having a normal olfaction. So if you're an individual who has LRRK2 PD and a reduced sense of smell, you're more likely to be SAA positive. If you are an individual with LRRK2 PD and a normal sense of smell, then you're more likely to be SAA negative. And the second interesting conclusion was actually came from two observations. The first, that alpha-synuclein SAA is negative in the vast majority of LRRK2 and GBA non-manifesting carriers, suggesting that the presence of alpha-synuclein aggregates in the CSF is not a lifelong trait, but rather acquired at some point relatively close to the disease. And the second was that alpha-synuclein results were positive in prodromal PD without dopaminergic imaging abnormalities in a substantial number of cases, while the converse was much less common, indicating that alpha-synuclein SAA may be a very early indicator of synucleinopathy. And so this is very interesting and allowed us to start placing alpha-synuclein pathology, dopaminergic degeneration, and clinical manifestations in the continuum of PD progression to establish different stages of PD, very much like the ATN framework for Alzheimer's disease. And it also allows us to create a biological definition of PD. Also, further validating, slide is not moving. There we go. Also, further validating the SAA results, we looked at some examples from brain pathology. We don't have too much here, but we have a few individuals in the PPMI who have come to postmortem. And if we look at those individuals, there is only a very small number of cases, but there is an 100% match that if you're synuclein negative in SAA, you don't have synuclein pathology in your brain. If you're synuclein positive in SAA, you do have synuclein in your brain. And this is also true in another small study supported by the Fox Foundation from the lab of Eduardo Tolosa and his colleagues in Barcelona, who was published last year in Movement Disorder. So this really gave us more confidence to make the leap that synuclein SAA does reflect synuclein in the brain and can perhaps be utilized to identify disease biology in these individuals. And furthermore, it does appear that the LRRK2 group represents at least two different biologies, one with synuclein pathology and one without it. We went further looking at subcohorts of individuals at risk of developing Parkinson's disease, namely individuals with hyposmia and REM sleep behavior disorder. What you have here is a representation of the individual SAA with the blue circles representing SAA positive individuals and the white circles representing SAA negative individuals. The x-axis show you the UPSEED percentile, and with this vertical line representing the 15th percentile, below which individuals are generally considered hyposmic. On the y-axis, you have a measure of DAT binding with this horizontal line defining the 65th or the 65%, which is generally accepted to indicate, or below 65% is generally accepted to indicate dopamine degeneration. And what you can see is that in both groups who have not clinically defined Parkinson's, most of the individuals are SAA positive. And among those who are SAA positive, they are also hyposmic, and which is a very strong association we identified from this study. And interestingly, not all of them have that deficit, suggesting that maybe there is a temporal pattern here where synuclein comes first and then that comes next. And so as a follow-up from these results, we can start thinking about this temporal paradigm. And this actually has also been replicated in other data sets like the PAR study. So why is this important? Why it is important to rethink Parkinson's disease in biological stages? Obviously, there are major challenges for development of PD therapeutics. PD is, of course, a progressive disorder, but with a very slow rate of decline within the time frame of a clinical trial. It is a very heterogeneous disorder with several subtypes. some of which with likely different etiology, disease course, and response to therapies, despite the fact that in many clinical trials, people try to recruit newly diagnosed individuals. There is a significant portion of the population that will need to start taking medication soon after the diagnosis, creating other co-founding factors for the trials. And of course, as we know, degeneration begins well in advance of the onset of clinical symptoms. And by recruiting individuals with clinically diagnosed Parkinson's, we're not really intervening early in the disease process. So we turned again into the PPMI study to attempt a paradigm shift for Parkinson's disease that has been primarily driven by our ability to measure synuclein pathology in life using the amplification assay. This led us to what we think is a new definition of Parkinson's disease, and I will address that in a moment, but also allowed us to test a new framework for how we envision the progression of PD to occur over time. And the goal of all of this, continues to be to try taking advantage of these efforts to develop ways to improve and expand therapeutic development in PD. So this is what we believe is our paradigm shift. This is a classical slide representing the natural history of Parkinson's disease. And of course, most of the information we have is on the right side after the onset of symptoms. And what we're proposing is to further define what happens in this left side prior to the onset of symptoms, where we might be able to intervene and shift our thinking so that what has always been a diagnosis based on clinical symptoms would be a biological diagnosis based on a biological definition of the disease. And this would be evident in this slide. We now have more information from genetic status, but more importantly, from these two key sets of biomarkers, the synuclein amplification and dopamine transport imaging, that we believe can define this illness biologically at this stage prior to the onset of the classical symptoms. And so what we're proposing specifically is that Parkinson's disease is defined by the presence of these two specific biomarkers. Now, the biomarkers we have today are the synuclein amplification assay in CSF and the DAT scans. These need not to be the only biomarkers we'll be using in the future. It won't be. And so this has legs to improve as new biomarkers and tests get validated. But the concept that these two current ones are is that they are going to be sufficient to define this disease. And importantly, we now have the ability to identify and define the disease prior to the onset of symptoms, and we can therefore develop therapeutics to intervene in that time frame. Another important consideration as we apply this biological mantra is to think that a single biology may give rise to multiple clinical phenotypes, and that is okay. And also that the same clinical phenotype may be defined by different biologies, which seems to be what we're seeing with the LAR2 group, as I mentioned before, one being synuclein negative and the other being synuclein positive. But all of this really led us to the opportunity to apply a staging framework for PD. And the way we have set up this is restricted to neuronal alpha-synuclein disease. That's why my title, the change in my title. So multiple system atrophy is for the moment out of scope. On the other hand, since we're defining the disease based on the presence of neuronal pathology, we believe that Parkinson's disease and dementia with Lewy bodies are really the same disease, and we should be thinking about this as a single disease entity with different phenotypes. I'm going to show you the staging system in a second, but it basically has three different components. The first one being genetics, the second biomarkers, and the third measures of functional impairment. And we try to integrate those three domains to develop the staging system. The staging system is here. I know this is a very busy slide, but I'll try to go over it at a very high level. I want to point out that the top on the left, you can see a risk assessment with RL and R age. These are not really part of the staging, but are here to recognize individuals who have genetic variants with lower or higher risk of developing the disease. The actual staging system begins below that blue line at stage 0, which is really reserved for very specific kinds of genetic mutations, which have a very high penetrance for developing disease and are almost certain to develop synuclein pathology. The classic example being the familial cases with synuclein mutations, and which takes me to stages 1 and 2, who really reflect biomarker-based stages without any evidence of functional impairment. Stage 1 with no clinical symptoms at all, and stage 2 with mild or subtle symptoms, but without any functional impairment. And then stages 3 to 6 reflect increasing severity of functional impairment over time. At the moment, the biomarkers remain static from stages 2B to stage 6. But of course, we expect and hope that this platform will improve and evolve as we develop better biomarkers that can reflect the severity of illness, which is something we don't have available today. This is simply the same slide, but without all the wording, to make the point that we want to make this a simple process to facilitate the community in utilizing this in the future. To help understanding how this would apply in the real world, how would real humans being staged? We have here just a few examples. A 52-year-old individual with a GBA mutation, with normal SAA and DAT binding, he would be on this RL category. And you can see that the old terminology is a little loose: pre-manifest, preclinical, non-manifesting, at risk. So we're trying to sort of consolidate in a more objective way the classification of these individuals. An individual with RBD who is SAA positive and has an abnormal DAT but has no functional impairment would be in 2B. And they would be what typically is classified today as prodromal Parkinson's disease. And a 60-year-old man with abnormal SAA and abnormal DAT with Parkinsonism and slight functional impairment would be in stage 3, and they would sort of be the typical de novo PD patient that would generally enroll in a clinical trial today. So this is to give you some sense of how this would generally play out. This is another way, more graphical, of representing the staging system, similarly to what has been done for Huntington's disease. And this is to acknowledge that in some ways we're also learning from others. and that this is not something isolated for Parkinson's disease, but that similar processes have been generated in other neurodegenerative diseases like Huntington's disease and Alzheimer's disease. And so I want to wrap up. It's actually been quicker than I thought, but I want to wrap up with a concept and take a message of this exercise. Parkinson's disease and related disorders today have many different features and faces and definitions. But if we define it by its biology, then all these syndromes arise from that same biology of synuclein pathology. And, they are essentially the same disease. Of course, that doesn't mean that in clinical practice, people are not going to be called Parkinson's disease or dementia with Lewy body, etc. But it does mean that as we think about therapeutic development, we might want to think about this in a different way and enable us to define therapeutic development based on disease biology, because ultimately, new drugs will target that biology. And so I think I've actually done. So thank you. It was quicker than I had imagined, but that means that we may have more time for questions and discussion, which is exciting. So thank you.
Thank you so much. And I will ask that you go ahead and leave your slides up in case we need to reference any previously. But at this point, we can take any questions. I'll start real quick with Dr. Devos left a question in the chat. The results of the diagnostic accuracy, he wanted to know if this was on the de novo PD.
Well, this is on the de novo PD. That is correct. So so if I go back to the slides with the learnings of here. Oh, yeah, this sensitivity and specificity was for sporadic PD versus healthy controls at baseline, and PPMI, was mostly enrolled individuals early in disease, obviously to prospectively follow them over time. And so at baseline, these were essentially de novo PD. I must say that when we looked at the prodromal populations, namely RBDs and hyposmics, the percentages of sensitivity and specificity were pretty much identical. So again, suggesting that synuclein is something that comes up very early in the disease process.
Thank you. And I see Dr. Moon has his hand raised.
Yes, I actually have two questions. So this synuclein assay actually uses a CSF. So are there any other researchers looking at the blood biomarkers? Because taking CSF is kind of trickier than the taking blood, right? So I was just wondering about that. And then the second question is, why this synuclein appears right before the disease onset. So it's not like a lifelong accumulation. So it kind of appears in the beginning of the disease. So I was actually, is there any mechanism already discovered related to that phenomenon?
Absolutely. Two great questions. The first one is, you really nailed it in a sense that this is one of our priority goals now at the foundation, is to help with, to mostly thinking about the patient population. Like you said, having a lumbar puncture is not something to take lightly. And so it is a big effort for us to try to now transition this assay from a CSF-based assay into, some other peripheral matrix. Like I said in the beginning, we know that alpha-synuclein, pathological alpha-synuclein, actually travels or is present throughout the body. There are multiple evidences of that in the literature. We think, blood is definitely the gold standard. And we actually earlier this year also had publication in Nature Medicine from a group in Japan claiming that they were able to run this test in blood. We are already collaborating with this group, so they are receiving PPMI samples, and so we'll see if we're going to be able to replicate these early findings. If we look more broadly into the literature, the peripheral matrix that seems to come up on top as number of publications and some agreement between different groups is skin. Obviously, skin is we also have neuronal fibrils in skin, and they are known to accumulate alpha-synuclein. And so we're actually in the process of also validating. So we started, because of that evidence, we started collecting recently skin biopsies as part of the PPMI study, and we're starting that now, sort of processing these skin samples to look at SAA status and see how that compares with the CSF status, and hopefully, I would be hopeful that by earlier next year, we'll start to have the first results to test the possibility that skin could be an alternative matrix to CSF. Now, I also wanted to point what I think it's a really interesting concept is that the correlation between the CSF SAA status and olfaction was really, really striking. And so as we think of going to the general population and screening individuals in a more larger scale, which obviously wouldn't be possible with lumbar punctures, but at least as almost like a surrogate marker of SAA status, a simple smell test could indicate a higher probability of having synuclein pathology, and this is actually something that we're implementing in PPMI as we are thinking of expanding this study to look for populations early in disease. So we want to characterize the natural history of Parkinson's in its entirety, in its full continuum of the disease, and so we definitely need to go early, and so we already have implemented what we call a smell test direct. So people can now go into the PPMI website and based on responding some very simple questions, they receive for free a smell test at home, and then if the results are positive, then there can be a follow-up and potential conversations to enroll in the PPMI study more broadly. And this is something that we'll be launching worldwide, and so our estimations are that we'll be able to target something like 500,000 people worldwide, and have these information on smell as a potential surrogate for SAA status. The second question around why do we think synuclein happens so early in the disease? It's a very good one, and I don't have such a good answer as the first one. I mean, we know that alpha-synuclein is, it's part of the hallmark on how we classify the disease, right? Up to now, the confirmatory diagnosis for Parkinson's disease happens at autopsy when we look into the presence of these synuclein aggregates in the brain. So there's, and also from the genetic standpoint, if the synuclein gene is the strongest GWAS signal or the strongest GWAS gene for Parkinson's, and if you have certain mutations in the synuclein gene, you have almost 100% prevalence to develop Parkinson's disease. And so it really points synuclein as a central player in this process. And so what we think, and this is similar to other neurodegenerative diseases related to amyloid proteins, is that something happens in the cell where this monomeric functional protein misfolds and starts to aggregate. Exactly what that is, we don't know. It might be many different things. It might be an infection that results in high stress to the neurons and inflammation. It might be, as we age, and we know that aging is the major risk factor for Parkinson's, it might be that our protein control systems start to get defective. And so at some point in time, they're not able to cope with alpha-synuclein misfolding, and they cannot clear it, and it starts to build up. So there are really many different possibilities for this process to start. But what seems evident is that it plays a central role in disease pathology.
Thank you.
And Dr. Devos has his
Dr.
hand.
Oliveira, thank you so much for this wonderful presentation and also allowing us to ask questions. I think this interactive discussion is very much appreciated. I had two questions that are kind of completely independent from one another. The first question is that smell tests are not only indicative of Parkinson's disease, but are also under an investigation for other neurodegenerative conditions, such as Alzheimer's disease. Some work is coming out as we speak. I was wondering if there's specific smells that patients with Parkinson's disease are deficient in. So that was my first question. The second question is more focused on PD care and more specifically on PD rehabilitation, on how you think that the change in landscape to earlier biomarkers will change the clinical care of Parkinson's disease, and more specifically the rehabilitation of Parkinson's disease.
Mm-hmm. Two great questions. The first one, I have to admit my lack of knowledge. I don't know if there are certain sense that individuals with PD might be more prone to lose over time. I would have to double check. I know that the test that we use in this smell test direct program within PPMI is the UPSIT. So it's the smell test developed by the University or the Penn University. But I don't know exactly all the details on what that test measures. So happy to go get back to you if I'm able to get more information. The second one regarding the rehabilitation, it's a really good one, and what I think it might be helpful here is I'm going to give the example of exercise, which I think there's a big body of evidence that it is a very important and non-invasive I want to call it treatment, but care or something that we can do with these individuals that people believe might delay the progression of disease in somehow. And what I think is that prior to this staging system, we didn't have a good way to think about functional impairment. And normally we could think of, okay, this person was diagnosed, let's say, within the last 2 years, and we have this person that was diagnosed 7 years ago. But that doesn't mean that the person that was diagnosed 2 years, it's earlier in the disease process, right? Because we know that disease progression is very heterogeneous. And in fact, the person that was diagnosed at 2 years might have more functional impairment than the person that was diagnosed 7 years ago. And so what I think this framework allows us is to sort of classify these many different levels of functional impairment. And I didn't go, into that detail, but obviously, and also because this is still a subject of active discussion, exactly how are we going to measure and quantify functional impairment. But if we have individuals in stages of functional impairment, I think a good way to think about the impact of care and different strategies could be, for instance, how long it takes from one individual to go from one stage to the other, because we believe that now these clusters of individuals in stages will represent a more homogeneous population. So when we want to try how, let's say, cardiovascular exercise versus high intensity exercise, or two different diets, I think that this framework will now help to test these interventions in more homogeneous subsets of the population and provide a more objective way to measure the effect.
Thank
Thank
you.
you.
And we have another question. Monica Gross. Go ahead.
Yes, hi. Thank you for this great presentation and for all the hard work you've done. Just for what Hannes was saying about olfactory and specific smells, I know Parkinson's Foundation in their little insert about the early signs of Parkinson's, they mentioned smells, smelling bananas, dill pickles, and licorice. So that would be sweet, sour, spice. So it looks like it's a
Thanks.
range of
Information
scents.
for me.
Yeah. Sometimes the consumer facing stuff is easier to absorb. My question, and I know it's not the subject of this, your research in this presentation, but how would this link with the new discoveries through the university in Helsinki about the bacteria that's been found and located and identified as showing up significantly in prodromal stages and with Parkinson's? Is that another biomarker potentially, or how would that be treated?
Yeah, I mean, potentially, yes, if we can replicate that at a larger scale. I mean, there's definitely a lot to know in terms of understanding causes of Parkinson's and, you know, what could be useful markers. And you may have heard of these two ideas of brain first and body first Parkinson's as well. So these are all things that, you know, are not ready yet to be captured within this staging framework. But again, this is an open framework and it will improve over time. I think the presence of a bacteria, we know that some bacteria produce certain proteins like curlin that in various in vitro studies and in cellular studies, and I think even in animal models, have been shown to promote synuclein aggregation, and so that is a possibility that in a certain set of the Parkinson's disease population, that
Right.
might be triggered by an increase in a certain bacteria that, you know, through the infection can start the disease process in alpha-synuclein. But definitely, you know, needs to be replicated for sure in other cohorts. Yeah, and we'll have to see if, you know, we're able to have a standardized measure of that bacteria in order to be able to utilize that in the context of a large clinical system, either PPMI or in a clinical trial.
Thanks. And just another question, would is it possible for you to share your contact information so that we can stay in touch and learn more about, you know, the process? Is
Absolutely. How
in
would
the
I
chat?
do that?
In the chat, I
Can you,
guess.
or in the
I
chat,
can go ahead
or
and if
Thanks
you
so
want to
much.
put that in the chat, we won't put it on the screen just
Mhm,
because this
mhm.
will be posted later too. But yeah, in the
Right,
chat would be
right.
great.
Because this was such a lot of great information and absorbing it will take a little while.
What I'm not seeing is the chat. Oh, it's
Um,
here. I
okay,
see it.
perfect.
Chat,
And real quick,
yes.
as you're doing that, I'd like to throw in one question of my own. Looking at the slide that's presently up, the stages 0, 1A, and 1B, where you're not seeing those clinical signs and symptoms, so that's that preclinical stage. Um, in another neurodegenerative condition, Alzheimer's disease, there's been so much push over the last 5 to 10 years for early diagnosis and better diagnosis at the preclinical stage. However, that wasn't necessarily matched with a course of action for those that are better identified. So we now have the ability to tell someone that you're not experiencing the symptoms of Alzheimer's disease, but you're on that trajectory, or we have, you know, the evidence that that is to come. And that's a tricky spot because those people are left with very little that they can do.
Yeah.
So I wonder, is that the same for Parkinson's, or is the organizational infrastructure there to guide preclinical patients if we have the diagnostic capacity?
No, that's a fantastic question and something that, you know, we'll need to figure out with many stakeholders in the field exactly, you know, how, and also from an ethical standpoint, like we'll have to have very, you know, very strong science-based evidence before going to a person with a test that has no symptoms whatsoever of Parkinson's, and then, tell that person you actually have Parkinson's disease.
Right.
It's definitely, this we'll need to reflect on this. But from the perspective of clinical development, it's a huge opportunity. I go back to this idea that, all the clinical trials ongoing today that are mainly targeting what we call de novo PD, they're not intervening early. And it may be, it certainly, it is much more challenging to stop or slow the progression of disease at a time where the damage is already so large in the brain. And so I think we can't ignore this opportunity. If we just think on that perspective, obviously, if we're able to transition this test from a CSF matrix to a more peripheral matrix, we could almost envision a world where, people in their 50s would get, an SAA test as part of their regular annual checkups, and might be diagnosed with this. But I agree that there needs to be, a framework of counseling. Perhaps this could be more widespread available when there are already something we can do with that, like either prevent drugs for prevention in the pipeline, so that people can do something with it. Okay, I don't have any symptom of Parkinson's, but I have this positive test, and I know that there are 1, 2, 5 clinical trials testing a prevention drug, so I might be interested. So that's something now that I can do with this information. But I agree that just giving this information cold and not be able to do anything with it is something that needs to take some consideration.
Certainly. Thank you so much. Are there any final questions for Dr. Oliveira?
So are you based in New York City or?
Correct.
Yeah.
Okay, I'm going to go ahead and stop the recording here.